Uh, my name is Dr. Radha Krishna. This is a topic on neurodegenerative diseases and its relationship to intestinal bowel diseases. And in the case studies, we are supposed to identify the introduction to neurodegenerative diseases is association with inflammatory bowel diseases. And the second point is to expected to identify the underlying pathways, mention how it contributes to potential new target therapies. And the third question be students are expected to elaborate in detail each pathway with emphasis to at the molecular levels with appropriate figures or graphic illustrations on their own. The other one is the students expected to mention the molecular target associated with the above area of interest. Students are also supposed to give the end a conclusion of the summary of the whole topic without repeating the earlier mentioned report. Inflammatory bowel disease and neurodegenerative disorders like neurodegenerative illnesses, including Alzheimer's, Parkinson, multiple sclerosis, with a cognitive and motor deficiency, being destroyed the neural structure and the function. These illnesses are difficult to treat because they are complicated genetic, environmental, inflammatory etiology works in tandem. The newer research linking problems of inflammatory diseases like Crohn's disease, ulcerative colitis. Neurodegenerative illnesses and IBD are linked. The highlights the gut-brain axis, the bidirectional communication with the network of CNS and GIT. The gut microbiome varies collection of microbiome can sustain the gut health, and the systemic equilibrium can influence this link. So in IBD, dysbiosis of the gut can make cause chronic inflammation in the gut and other organs of the brain. Neurodegenerative diseases, intestinal bowel disease, shares inflammation. So the word is the inflammation. Chronic inflammation in intestinal bowel disease caused by abnormal immune response of the gut microbiome can cause serious systemic repercussions. Systemic inflammation can damage CNS, causing neuroinflammation, a fundamental characteristic of neurodegenerative disorders. The immunological, endocrinal, neurological network that connects to the gut and the brain are increasingly thought to play a role in the development and progression of intestinal bowel diseases and neurodegenerative illnesses. Better diagnosis and treatment of IBD need to understand the complex link between neurodegenerative illnesses and IBD. This relationship emphasizes on the necessity of the comprehensive approach to patient's care, considering the gut and the brain health together. In context, studying the gut-brain axis, chronic inflammation, gut microbiome might help to mitigate these debilitating diseases. The pathophysiology of Parkinson's disease. The Parkinson's disease is a progressive neurodegenerative disorder characterized by primarily motor symptoms such as tremors, rigidity, bradykinase, slowness of the movement, postural instability. The pathophysiology of Parkinson involves a complex interplay of genetics, environmental, biochemical factors leading to degeneration of specific neurological population, primarily in the substantia nigra. The genetic component can be the mutation of a nucleosine, SNCA, or Parkins or PARX2, DJ1, LURX2, and causing the protein to degeneration and mitochondrial function and oxidative stress. The genetic variation and the polymorphism risk the genetic predisposition of the environmental factors. If you look at this diagram, if you look at the gut producing inflammation or inflammatory disorder, the gut infections can cause LRT2 or it's a leucine-rich repeat kinases 2 that causes neuroinflammation, and this inflammation can trigger an immune cells to be spread to the brain area and cause a neuroinflammatory environment for the dopaminergic neurons in the substantia nigra to cause it to have neurodegenerations. Some of the methods for latest techniques of improvement of Parkinson's is involving CRISPR. Genetic editing allows precise genetic editing which targets mutations such as uh, synuclein cells, PARX2, LAX2 genes. These gene regulations of the CRISPR explores the upregulating or downregulating of the genes involved in the pathogenesis of Parkinson's. The other targeted therapies are alpha-synuclein targeted therapies, like targeting, if you look very carefully, it will be therapeutic or immune therapy, immunotherapy, or receptor blocking therapies. If you look very carefully at the bottom part, this is one of the latest techniques used for helping to overcome Parkinson's. Rifampicin, FDA-approved drug, has increases the autophagy levels by inhibiting the mTOR pathway, which reduces the dopaminergic cell death. Alpha-nuclein is a protein that aggregates in the brain, and using immunotherapy targeting alpha-nucleic cell aggregation, the small molecule inhibit the to prevent the aggregation or to promote its clearance. So neuroprotective agents, these therapies are aimed to slow down or to stop the progression of Parkinson's diseases, like NRF2 activators, which is one of the transcription factors that induces the antioxidant response and protect the dopaminergic neurons from oxidative stress. More pronounced are the mitochondrial function enhancers, which are involved in the redox mechanism, especially in the electron transport chain and several other mitochondrial therapies. The other that's gaining traction is the personalized medicine, where bioinformatic inclusion and omics. The treatment is tailor-made to specific pathways, identifying the specific biomarkers to effectively individualize therapies. Advanced drug management delivery system, where innovative drugs delivery system involve the nanoparticles or intranasal delivery. Both are non-invasive and bypass the blood-brain barrier, can be an effective can be given in effective therapeutic dosages. Addressing the root cause, like disease-modifying therapies, addresses the underlying causes rather than the symptoms. An example is the leucine-rich repeat kinase 2 or LUX2, which reduces the kinase activity and thought to be contributing to the neurological damage. Here's a sample where they use the leucine-rich repeat kinase 2 NSAID to inhibit NSAID, which prevents the mutation of the LUX2. So it reduces the inflammation at the neurons in the brain. This is another method where antisense oligosaccharides, these molecules can bind to the RNA, modifying the potential to reduce the harmful effects of the proteins as shown above. If we can see the DNA on the above, and when they convert to the RNA can convert to the protein. There can be an altered protein, or there is no protein synthesis in the first place. So this way it protects the person from Parkinson's diseases or advanced cases, or reduces symptoms. Genetic mutation of PARX, UCHL1, DJ1, PINK1 causes unfolding of protein response or UPR dysfunction, and as well as mitochondrial dysfunction and oxidative stress, resulting in neurodegenerative changes. If you look very carefully, you can see that the above goes into the mitochondria and causes reactive oxygen species and causes massive damage. As a result, it causes neurodegenerative changes. One of the other causes of damage is the mitochondrial dysfunction. There is a reduction in the activity in the complex 1 of the electron transport chain in the substantia nigra, impaired ATP production. There is an impairment of mitophagy, which causes the damage to mitochondria, selective degradation, leading to mitochondrial dysfunction and cellular response in apoptosis. There's another problem where there's a loss of dopaminergic neurons. The hallmark of Parkinson's disease is a selective loss of dopaminergic neurons in the substantia nigra compacta. The loss of this neuron results in decrease in dopamine levels. Dopamine is the critical neurotransmitter involving the regulatory movement, and deficiency disrupts the balance between the excitatory and inhibitory pathways of the basal ganglia. The hallmark of Parkinson is basically Lewy bodies and the alpha synuclein aggregation, intracellular inclusion, also known as the Lewy bodies, are the pathological hallmark of Parkinson's disease. Alpha nucleic aggregation into the insoluble fibers plays a central role in the pathogenesis of Parkinson's. So we can actually see the fibrils and they become insoluble, and as a result it forms the Lewy bodies. This neuroinflammation, microglial activation, and the residing immunocells releases pro-inflammatory cytokines and reactive oxygen species, which will exacerbate the neurological damage. Astrocytes also exhibit functional changes and contribute to neuroinflammation and impaired neural support. Oxidative stress, the impairment of mitochondria leads to oxidative stress, high metabolic rate, auto-oxidation, and leads to neurological damages. So in the development of new drugs, and the promising method and the drugs is Ongentis, and it's an FDA-approved drug. It's a catecholamin methyltransferase inhibitor, works along by prolonging the effect of levodopa. Nurance is another drug, FDA approved. It's an adenosine A2A receptor antagonist. The other drug is the Imbrija, which is a levodopa inhaler protein powder. It inhaled from the form of levodopa. These innovative approaches and the future direction will be on the gene therapy, cell-based therapies like stem cell, inducible pluripotent cells, neuroprotective therapies like mitochondrial dysfunction and oxidative stress. One of the most important connection between the brain and the gut in the case of Parkinson is the gut microbiome. So the gut microbiome influences the neuroinflammation, the neurotransmitters or transmitter synthesis, mitochondrial function, intestinal barrier integrity through gut-brain axis, where they contribute to the development and progression of the diseases. If you look at this diagram in the bottom part, the shikimate will ultimately result in ispronoid aromatic amino acids like phenylalanine, tyrosine, tryptophan, folate cofactors. Phenylalanine converts to tyrosine and three breakdown products called tyrosine, dopa, and melanin. What's the relationship between this and the Parkinson's? Here's a pathway I'm showing where phenylalanine converts to tyrosine. Phenylalanine also converts to dopa. This is where the dopamine is produced in the brain, and the other part it can be tyrosine converts to thyroxine. So the most important part of thyroxine conversion is the dopa, which is a central role in Parkinson's. Let's look at the kynurenine pathway. The tryptophan converts to kynurenine, and then it converts to quinolic acid, and this is where the Parkinson's affects the Parkinson. So kynurenine is neuroprotective, and quinolic acid is neurotoxic. This is where it all gets at the gut level, and this is where the conversion we have to carefully calibrate. So if you look at the kynurenine pathway and the quinolic acid pathway, if we were to do a ratio between the kynurenine acid and the kynurenine, or the quinolic acid and kynurenine acid, using a spectrometry, then we can calibrate what is going on in the gut, because gut bacteria produces all these products like tryptophan. So this is where the pathway comes in, and the other one will be the acyl carnitine, where it is produced in the intestine, and this can be also be measured by spectrometry. So for Parkinson's, the relationship between the brain and the gut, and neuroinflammatory disease of the gut can influence the neurodegenerative changes in the brain. So the bacteria specifically involved in this, especially we can use bioinformatics for critical role to identify and characterize these bacteria. The role can be used by computational tools. Biological data allow the analysis of large-scale genomics, transcriptomics, proteomics, metabolomic data to identify if it's identified. So when we do this, we can identify the specific species and their functions, exploring different environments, including the human gut, using the next generation sequencing, metagenomics, microbiome analysis using the humane, or it infers as the Kyoto Encyclopedia of Genes, and understanding the higher function and utility of the biological system. About half of the dopamine is produced in the gut. It communicates into the brain via the vagus nerve. Certain bacteria in the gut produces dopamine, like Bacillus subtilis, Lactobacillus plantarum, or Escherichia coli. Meanwhile, the mitochondrial biomarkers like acyl-carnitine using a spectrometry, uh, we used to identify them. So Lactobacillus rotary, Lactobacillus plantarum, and E. coli produces CoQ10, which is part of the mitochondrial dysfunction or oxidative stress. These specific bacteria can be placed in entry-coated capsules to prevent digestion from destroying the stomach bacteria in the stomach, where we can add between 10,000 colony functions or CFU to 20,000, which can be consumed daily. And if we look at the previous market, they were already doing the gut bacteria transfer or implantation. So we can make it to a different approach using capsulization and protection and identify the specific bacteria, and we can also target Parkinson's disease. So if you look at the neurodegenerative diseases associated with IBD, if you look very carefully, this diagram shows the microbiome influences the brain via vagus nerve and the non-vagus nerve. So it is dopamine receptors are present in the gut, and dopamine is produced in the gut that can influence via the vagus nerve to the brain. In conclusion, apart from the FDA-approved drugs, like I've mentioned previously, including the nasal uh, dopamine inhalers and many other drugs, there's also a gut methodology of improving Parkinson's by overcoming neuroinflammatory inflammation of the brain via the gut microbiome. So we can have very specific targeted microbiome like Bacillus subtilis, Lactobacillus plantarum, Lactobacillus rotary, E. coli that produces CoQ10, tryptophan, phenylalanine, tyrosine, dopamine. Very specifically is dopamine produce chironinin, that is a neuroprotective, putting together as an additional role to fecal implant. Dr. Radha signing off.